All right, this is Dr. Brice Semmens, leader of the Grouper Moon Project. Brice, we got a boat loaded with different technologies here. Tell me about some of it.
Yeah, so we're starting here with, we're out here at the Grouper hole and we're fishing for grouper to try and put acoustic tags in. First piece of technology I'm going to tell you about is a hook. Now, this you attach to the end of a line, usually with some weight, and you put bait on it.
Really?
Yeah. The bait on the hook is what the fish bite. Because the hook is attached to a line, you can then pull the fish up to the boat.
I think we got that.
Okay.
Can we move up to a little more modern technology?
Absolutely. Let's do that.
Let's do that.
All right.
All right. What's this right here?
That is a set of scuba gear. You have a tank. This tank has compressed air. Allows you to have air for breathing underwater for long periods of time, maybe 45 minutes or so. It's got a regulator on it. This regulator takes the highly compressed air and turns it into the level of compression you need in order to breathe, just like you'd be breathing on the surface. And finally, this piece right here, this is essentially an inflatable bag that you have on the back between the diver and the tank, and that allows you to maintain neutral buoyancy. So you're not sinking and you're not floating. We use this gear to go down, observe the fish, collect data, and do all the work we need to do down below.
All right, so we've got divers looking at fish, we're catching some fish, and let's see, what else are we looking at? Oh, there's a little gadget over here by the side of the boat.
Yep. This is...
What's this?
This is a Vemco VR100. So after we're done tagging the fish with an acoustic tag, which is a small piece of electronics, that acoustic tag emits a sound at regular intervals that this piece of equipment has a hydrophone and can translate that sound into a uniquely identifiable signal for each of the tags that we implant into fish. And by doing that, we can keep track of each fish that we've tagged individually and uniquely.
Okay, so we're listening for fish too. And let's see, what else we got? Oh, well, now we're moving up the ranks into pretty high-tech stuff, it looks like right here.
Yeah, so
what's going on with that?
This is, the reason we're out here is because this site is where Nassau Grouper will spawn. They'll release their eggs into the water column, and on nights of peak spawning you'll get thousands and thousands of individuals releasing eggs all at the same time. One of the things we're interested in is where are those eggs going and how fast are they getting to where they're going. So we use this piece of equipment, which is a plankton net, which we typically use to catch things like eggs and larvae in the water column. In the middle of it looks like what's a missile. This is actually a flow meter, so it keeps track of how much water the plankton net is sampling. And then below, at the very end, right before the cod end, or the place where all the plankton larvae end up, we've got a set of microscopes and strobes. So each one of these is a microscope, and each one of these is a strobe to light up that microscope image. And we capture images in real time, about 10 every second or so, as this plankton net is towing through the water. And by doing that, we can not only catch the eggs and larvae, but we also get a record in real time of where that egg was caught, at what depth it was caught, and when it was caught. And through those pieces of information, we can reconstruct, by towing across where the Nassau Grouper have spawned, we can reconstruct where those eggs are and where they're going through time.
Okay. And I got one more thing to ask you about, or well, two actually. This is the first one. What do we have there?
Yeah, so we talked about going on a dive with scuba gear. This is actually a Gopro camera, or a small video camera, and it's affixed to a housing that inside maintains 4 different lasers. And so we can switch on a knob and those lasers will turn on, and they're perfectly parallel, which means no matter how far or close I am to an object, the 2 red dots that those lasers create are always exactly about 25 centimeters apart. And so I go down, I'm videoing, I get those 2 lasers parallel and on the side of a fish, and I capture that image with 2 red dots of the fish that I know are 25 centimeters apart, and I can subsequently take that video back to the lab and use the fact that I know 2 dots are 25 centimeters apart to ultimately estimate the total length of each fish that we laser.
All right, and one more gadget up in the cabin up here. We got 5 or 6 tubes full of electronics and things like that. What's that?
Yeah, so these are, I said before, one of the things that we're interested in is figuring out where the larvae go. These are part of a large set of autonomous underwater vehicles that we deploy when the fish spawn, and we put these things into the spawn cloud. And these and some other floats that we have that you're not looking at right now allow us to track the oceanography and spatial distribution of the spawn after it's released.
Well, these are fancy.
Very fancy.
Very fancy, but heavy duty, I assume, because they got to sit in the ocean for days tracking larvae, huh?
That's right. Yeah, they'll be in the ocean for up to a couple of days. And what these particular objects do is they emit a very high frequency and very loud sound that can be heard up to several kilometers away. And because we have multiples of these that are making those sounds, we can use their location with GPS and the sound frequency that they're using in order to triangulate other AUEs, or autonomous underwater vehicles, in real time as they move through the ocean.
And everywhere I look, I see new stuff. What's going on with the these big sock-looking objects on the right here?
Yeah, those are what are called Pacific gyre drifters. The black orb on top is attached by a tether to these pieces of hoops and fabric below. We throw that whole unit over water, into the water. The black orb up top has a GPS and a satellite phone, and it's constantly floating at the surface, broadcasting its location, and the sock itself hangs down below, and it's carried with the currents. So we have that sock so that the black orb doesn't get pushed by the wind and waves, but actually is dragged by the sock below with the current. And by doing that, we can keep track of that water below the surface where the eggs are spawned into. And this allows us to track where the eggs go as well.
So it sounds like all this technology puts a big puzzle together about how many fish there are underwater, where they spawn, where the eggs go after the spawning happens, and all this technology is needed to figure all that out.
That's right. So we're interested in figuring out how many fish are out there, where they exist throughout most of the year, how many come to the spawning site, and after they spawn, we're interested in figuring out where those eggs and larvae ultimately end up. And that's an important question because we need to know whether or not the fish that are spawning here are responsible for the new recruitment or the new juvenile grouper that are recruiting to the local reefs.
All right, Brice. Hey, thanks for the technology tour and information on the project.
All right, thank you.
Go Grouper Moon.
Go Grouper Moon.